right. Okay, good afternoon everyone. Please confirm online people. Am I audible to everyone? Online people, please confirm, am I audible to everyone? Okay. Okay, not a problem. Okay, it's playing or not, that's what we want to know. Good afternoon. Monica, just confirm, am I audible to you? Quickly confirm. Okay, perfect. Okay. So, I'll be discussing the TS set 2024 paper, the paper which was happened last time, and then in the next class we will be discussing the 2023 followed by 2022 paper. Okay. Now, everyone try to analyze the questions very carefully, and we have to understand, what is the chapter weightage, which means from which specific unit, how many number of questions were appeared. Now, all of you remember the most important statement when we consider the question paper of 2024, not even one question also, it is not at all from experimental, no question from analytical, right, and hardly one or two numerical questions are there. So then how the questions are framed? All the questions are mostly statement, okay, direct, and some are application based, right. So only these are the three ways how the questions were asked in the exam. Okay, so now let's start the question number 1. So please do not worry that you have to, do not think that you have to answer every single question. Okay, remove that thought from the mind. Suppose example, if you are solving the 100 questions, make sure that at least 70 to 75 questions you give the right answers. Okay, do not expect that we have to answer every single question. Okay, all right, let's start the first question. Everyone, all of you online people, please give the answers quickly. Slowly, huh? All right, everyone online people also please give the answer. The question says that which of the following statements are correct. Of course, the statement 1 says that polarity of water makes it an excellent solvent. Suppose if you see the structure of the water molecule having 2 hydrogens which are partially positively charged and with 1 oxygen which is partially negatively charged, right. Of course, water having the great ability to act as a universal solvent due to its polarity, and that too because of the presence of hydrogens as well as the oxygen. Of course, water has high tensile strength. There is no doubt, it is absolutely true statement. See, tensile strength is basically explaining about what? Cohesion only. If you quickly recall xylem, how water molecules can be moved within the xylem with the help of cohesion as well as the adhesion. So what is cohesion? How one water molecule is going to combine with the another water molecule. This is what we generally call it as the cohesion. So definitely water has a high tensile strength. Because of that only, it shows the cohesive properties. Water has high dielectric constant also, which is nearly 80. So these are very straightforward questions. The answer is, option 4. All are correct, right? Now, they are easy questions. Next, second question. Everyone please answer the second question. The peptide bond in a protein is, and I believe everyone remember what is peptide bond first of all. The bond which is formed between amino group and carboxyl group of 2 amino acid. I believe everyone quickly recall. So if you look at the amino acid, right side it is having carboxyl group and left side it is having the amino group, 1 hydrogen and 1 R group. And this will be the another amino acid, carboxyl group, 1 hydrogen and R, correct na? Whatever the bond that is formed between these two groups, We already read what is this reaction? Condensation reaction, where there is a loss of the water molecule. Then what do we get? We get a CONH bond, correct now, which is called as the peptide bond. Always remember friends, peptide bonds are what? Always planar, correct now? They are always in what form? Planar. And most important, every student has to remember all the peptide bonds exhibiting in what conformation? Trans conformation only. And the question is very clear, why they cannot exhibit in the cis configuration? Of course, they cannot exhibit in the cis configuration due to the steric hindrance, right? Due to the steric hindrance, they cannot exhibiting in the cis configuration, they always exhibiting in the trans configuration only, correct na? So it's a simple straightforward question only. These are all statement based, no analytical or logical. Choose the correct statement about peptides in the RC plot. This is also what? Informative only. We all read the RC plot. RC plot generally explains about what? Angles. It is generally explaining about the dihedral angles in a polypeptide. This is simply about the RC plot. So choose the correct statement about the peptides in the Ramachandran plot. Peptides that are unstructured will have the backbone dihedral angles in the disallowed region. No, they should have a specific angles. It is not possible to conclude whether the peptide adopts a helical or a beta sheet conformation. It is possible. It is possible we can say whether it can acquire the alpha helix or whether it can acquire the beta pleated structure. See, I told you clearly 99% of the proteins they contain alpha helix also, beta pleated also in their secondary structure, whereas some of the proteins, only some of the proteins, they only contain alpha helix or they only contain beta pleated, but they can definitely acquire a position, right? So this statement is also wrong. The occurrence of the beta turn conformation in a peptide can be deduced, which means whether the protein can exhibit alpha helix or the protein can exhibit beta turn, that we can easily analyze. So this is the most appropriate statement. The sequence of the peptide can be deduced. What do you mean by that? By using Ramachandran plot, we cannot analyze what is the sequence of amino acid. Means which is the first amino acid, which is the second, which is the third, fourth, fifth. We cannot analyze this by using the Ramachandran plot. What we can analyze whether it is acquiring the alpha helical fate or whether it is acquiring the beta sheeted plates, that we can easily analyze, right? Hence the most appropriate straightforward statement will be the option 3. Simple, okay. And see, this is, we can say 12th standard level question. Assertion: amino acid tend to be least soluble in water at their isoelectric point. And what is the reason? The net charge on amino acid is a function of the pH of the solution. And is it true or false? Assertion. Assertion is definitely true. And what about the reason? Reason is also true explanation. Correct now? An example, if you, let's quickly recap, amino acid contain carboxyl and amino group with one R group. Correct now? When we dissolve in the water, generally what happens? This loses this proton. Upon losing the proton, it is converted into what? Negatively charged. Upon gaining the proton, it is converted into the amino form, NH3. Correct now? So when it is possible? Whenever it is dissolved in the water. Correct now? So this is what here, it get both positive charge also, it will also get the negative charge also. So what is the net charge here? Net charge is what? 0. That point we generally call it as what? Isoelectric point. Isoelectric point is the point at which the amino acid exhibit in what form? Zwitter ionic form. So definitely assertion is a true assertion. The net charge on amino acid is the function of the pH of the solution. Absolutely true, this one also. So the correct option will be both assertion and reason are true, and R is the correct explanation for the assertion. That's the right statement. Then question number 5. Everyone answer online, people. Which of the following statements about bDNA structure is correct? Incorrect. Why it is wrong option? See, we should always remember number of purines equal to the number of pyrimidines. Correct, na? But what they are saying, now the amount of adenine plus thymine is equal to the amount of guanine plus cytosine. This is wrong, and we already studied that bDNA is the most commonly formed DNA in the human, right? And definitely it is a double helix. There are 10 base pair per one single ton. Correct, na? The double helix contain what chains? Anti-parallel chains. Separation of the two strands of the double helix require untwisting of the helix, right? So the incorrect statement will be the option 3. They are very straightforward statements, okay? Even 12th standard students also can answer. What is the answer for question number 6? Everyone. Others online people? What's the answer? See, glucose and mannose are epimers. Okay, now this time they may ask the question with respect to the galactose also. See, epimers, in very simple words, epimers means most of the structures are similar. They will be differ mostly with respect to one carbon, or the atoms present in one carbon are completely dissimilar or opposite orientation compared to the other. Suppose example, if you consider the mannose and glucose, how these two are differ with each other at which carbon? Second carbon. Correct, na? So both are completely arranged differently. In case of a mannose, the OH group is projected towards the left hand side, whereas here the OH group is projected towards the right hand side. Correct, na? Now if you, so definitely glucose and mannoses are what? Epimers. If you consider the glucose and galactose, we are considering what carbon here? Fourth carbon. So you have to remember definitely the carbon numbers. So here there is a similarity with respect to the fourth carbon, where the OH groups are arranged in a opposite orientation. So remember a common statement, mannose and glucose are epimers at the second carbon, whereas glucose and galactose are the epimers at the carbon number 4. Definitely try to remember the carbon numbers here. Very good, very good. Okay. This is also 12th standard question only. They are straightforward statement. Everyone online people, please give the answer for question number, what is this, 7? Online people. Try to eliminate the options. Everyone online people, what is the answer for the question number 7? Which one more? What is the answer? After a long time you are coming. You are alive. What's the answer? Mamata, what is the answer? See, of course, we know that we are having two fatty acids. One is saturated fatty acid and another one is the unsaturated fatty acid. We know very well unsaturated fatty acids are always increase the membrane fluidity, right? Because of the presence of a double bond, it makes the kink or the bend. And most important point here, depending on the presence of the double bonds, we further categorize it into different types. We call it as the MUFA and PUFA, right? Monounsaturated and polyunsaturated fatty acid. If they are having one double bond MUFA and multiple either double bond or triple bond more in number, we generally call it as PUFA. If an oil or a fatty acid, if it contains more MUFA and PUFA, which indicates that they are extremely healthy. Nowadays, whatever the vegetable oils that we get, they generally contain more PUFA and MUFA and PUFA only. And second point, all the unsaturated fatty acid at room temperature, they are acting as liquids, which are our vegetable oils. If you look into the saturated fatty acid, almost every single saturated fatty acid at room temperature, they are acting like what? Solids. So definitely saturated fatty acids having high melting point, is it? Whereas unsaturated fatty acids are having low melting point. So definitely unsaturated fatty acids contain the double bonds. Double bond in unsaturated fatty acids are mostly in the cis configuration. If they are in the trans configuration, bonds never become stable. To maintain the stability, they are always present in the cis configuration. In polyunsaturated fatty acid, that is PUFA, double bonds are almost never conjugated. See, if conjugated means they will arrange in this way, like a saturated fatty acid. Hence, they cannot maintain the fluidity. How always they will arrange? They will arrange in this way. So, which leads to what? A great gap between them. More the gap they contain, they can increase the fluidity very easily. So, they never be conjugated. That is right statement. We already discussed what is the ultimate goal of the function of the carbohydrates on the plasma membrane? Very good. So, which we generally call cell-cell recognition. For that, we require the carbohydrates. So, sphingoglycolipids, we already studied our nervous system is exclusively rich of glycolipid. So, what is sphingoglycolipids? Whereas sphingosine associated with the glycolipids, they are involved in biological recognition. Right statement. Even you can say sphingolipids are also involved in biological recognition. You remember next chemical, prostaglandins. I have told you in many lessons. What are these prostaglandins? Very good. They are the chemical mediators which are involved in the process of inflammation. Correct na? We studied, friends, most of the prostaglandins, leukotrienes, testosterone, estrogen, progesterone, they are all derived from cholesterol as their backbone. And that ring we generally call what ring? Cyclopentanoparahydrophenanthrene ring, which is generally how many carbon atom it is? 5-member ringed fused at. Correct na? So, number 1 statement is correct, number 2 is correct, 3 is also correct, 4 is also correct. What is the 5th statement they are asking? Prostaglandins, which are inflammatory chemicals, whenever they are showing their functions, mostly they use as intracellular messenger, which is cyclic AMP. Why this is wrong? Very good. Because we have studied, because this is what derivative? Cholesterol derivative. Cholesterol never uses the GPCR. It can easily enter into the nuclear membrane, into the nucleus. So, they will directly go and bind with the DNA domain, which is present within the nucleus. So, mostly they don't use any secondary messengers. There are some possibilities are there, but mostly they have specifically asked about the prostaglandin. They don't use any secondary messengers. Hence, option 1, 2, 3, 4 are the correct statements. So simple question. Everyone answer. Question number 8. I believe everyone remember Michaelis-Menten. What is substrate concentration derived as? What is the derivative? Substrate concentration. Km. Very good. What is turnover number? Kcat. Correct na? What is, when the enzymes become saturated? When it reaches complete Vmax. Correct na? What is half Vmax? Half of maximum velocity. So, can I say that enzyme at a Km value, it generally reaches what? Half of its Vmax, that we already studied. Now, every student please answer, what is the formula to calculate the Kcat? What is that? Everyone online people tell me, what is the formula to calculate Kcat? What is called Kcat? In other words, so what turnover number? Can I say that all the enzymes are already reached saturation point? Correct na? So Kcat equal to Vmax. How many enzymes reached Vmax? All the enzymes. We called as ET. Don't forget. Kcat we discussed now, Kcat equal to Vmax by ET. Means Kcat is the point where all the enzymes are already reached a saturation point. All the enzymes. There is no more enzyme is left out. Or all the active sites are occupied. So Vmax by ET. Please don't confuse, okay? Anyway, what is this question says that? Michaelis-Menten. Rate of enzyme reaction is independent of substrate concentration. It is dependent. Rate of non-enzyme reaction is proportional to the? No. Km is the enzyme substrate complex. No. Km is what? Km is the substrate concentration or the point at which enzyme reaches half of its maximum velocity. Right. So they are saying Michaelis-Menten states that enzyme substrate complex formation is essential in enzyme reaction. Of course, true statement. There's no doubt about it. So simple. Now, everything is there in PPT only. So simple lines. Everyone, please answer. Question number 10, online people. Which of the following statements are correct about retrotransposons? Retrotransposons means DNA or RNA? RNA transposons. Very good. Try it, everyone. What's the answer? Every student, we have two type of transposons. One is DNA and RNA. Correct, na? This is very clear. What we discussed, friends, when we consider humans or when we consider eukaryotes. Eukaryotes contain both DNA transposons also, RNA also. But which is most prevalent? RNA. When you consider the bacteria, bacteria contain majorly only DNA transposon. So can I give a common statement? Transposons are present in all organisms. Yes or no? So now the first option says that they transposes via RNA intermediate, which is retrotransposon. Correct, na? Remember everyone, what is this mechanism? Cut and paste or copy and paste? Correct, na? Which are copy and paste mechanism. Definitely there is an increase in their copy number. They contain genes for ribosomes. Transposons never code for any ribosomes. Do you think transposons code for RNA polymerase enzyme? They never code for any RNA polymerase enzyme. They are found only in bacteria. That is wrong statement. They may contain intron. Introns are very common, right? So only what is correct? Option 1 is correct. Option 5 is correct, right? Answer is 3. Okay. Clear, everyone? Everyone give the answer, online people. Don't be silent. This question is very simple, but you know. See, the question is very clear. It is accurately describes. Most of the students, only 2. See, this is what most of the people get confused here. This is a very good question among all. Of course, this statement we will neglect because facilitative diffusion is a type of a diffusion mechanism where there is no requirement of energy. Of course, you all people can answer this question very correctly because they use the ATP to pump ions against gradient. See, very tricky statement or good statement that they have given, it regulates movement. See, you have to understand, they are not saying that it is involved directly in the water movement. See, water can be transported via aquaporins, but what is this mechanism? Osmosis. Now, what is called osmosis? Movement of molecules from higher to the lower. What do we call it? It as gradient. Correct? That gradient can be created by using ATPases. You are getting the point. So in very simple words, sodium potassium ATPase pumps are required to increase the osmotic gradient of the osmolytes. We never ever say that they are directly involved in the transport of the water, but they regulate the water movement by creating the gradient. For creation of gradient we require ATPases. So originally this is the right answer, but when we consider this option, this is also a most appropriate option, but it is an indirect statement. But in a given options, they have given the 3 also. Definitely we have to choose that. They definitely require the energy, so option 4 can be eliminated. So what is the answer? Option 4, 2 and 3. So simple questions. Take your time and correctly give the answer, everyone. Others, everyone, online people, right or wrong, give the answers first. See these options also, 1, 2, 3, 4, so simple options, okay. Now anyway, now look at the point. Activation of heat shock proteins, also called as chaperones. Okay. Here they have given one point very clearly. All these mechanisms are responding whenever there is a stress. So now they are asking which mechanism will become active upon which stress condition, or what happens if that stress mechanism activated, what happens at that time. Suppose activation of heat shock proteins to repair damaged DNA, correct na? So DNA got damaged, definitely what happened? See, generally we remember, right, chaperones or heat shock proteins are required for the proper folding of proteins, correct na? Even HSPC is also involved in the DNA repair mechanisms also, right, especially in the SOS or BER repair, because what generally happen, due to the oxidation stress, I don't know whether you remember or not, certain oxidizing agents, anyone remember free radicals, I told you, OH minus, they generally changes the base conformation. At that time these repair mechanisms are active with the help of HSPs. So A will be the one, there only we can come to the conclusion anyway. Upregulation of antioxidant enzymes to neutralize reactive oxygen species. What are reactive oxygen species? Best example, H2O, hydrogen peroxide, this OH minus, these are all what? Free radicals. If we destruct the free radicals, how can we destruct? By releasing antioxidant. Best example of antioxidants, you can take a glutathione, correct now, which destruct the free radicals. So B will be the 2. Then C, inhibition of protein synthesis to conserve energy, correct now, which is what pathway it is? Metabolic pathway. Once we reduce the protein synthesis, definitely we can store abundant amount of the energy, because compared to the replication, transcription, translation require more energy. So if I reduce this translation, I can use more amount of the energy. Increased production of osmoprotectants to maintain the cell membrane integrity, especially when the plant cells are facing certain oxidative stress or water stress, especially in the freezing condition, cooling condition. They are going to release some chemicals named as osmoprotectants. We read it, proline, betaine, trehalose. We read in the stress physiology the same line. We call compatible solutes. Those solutes are only responsible for maintaining the osmotic balance during the oxidative stress. Simple. What is the function of them? Function of osmoprotectants is what? Maintaining the osmotic balance during the stress conditions, right? So this is the 4th option. So correct answer is what? Number 1. Simple question. This is also informative. Of course, it takes little time, that's all. And what I observed is, just common thing I am saying, out of 500 questions, if I consider all the question papers of 5-6 years, every single time the answers will be very straightforward. And what else I have observed? Suppose example P, Q, R, S they have given in a statement related questions. Almost all the options are correct. This is what I observed. Out of 500 questions, I observed more than 350 questions in the same way. And I also observed in more than 350 questions of assertion and reason, 99% out of 300, both assertion and reason are true. Okay, that is commonly observed, okay, but every time it won't be correct. And when it comes to the matching, I observed number 1 is the answer for most of the time. Okay, so this only happens in set exams. Okay, next question number 12. So simple it is. Slowly. So this is exception. Everyone. Peroxisome play a crucial role in the detoxification reaction. Is it? See, there is, tell me Mamata, what is the name of the enzyme that breaks down the H2O2? Catalase. So catalase is present in the peroxisome, which is involved in breakdown also, making also. Both making and breaking up H2O2 happens within the peroxisome. We also discussed peroxisome plays a very key role in the biosynthesis of very long chain fatty acids. What is the main function? To store calcium ions and regulate intracellular calcium levels? No. This can be done by smooth endoplasmic reticulum and sarcoplasmic reticulum. Right? So reason is the false, only the assertion is true. Answer is 3. This answer is also wrong. Okay. Out of 100 questions, definitely 10 to 12 questions are definitely wrong. Tell me Sandhya, what is called APC function? What is the major function of APC? Anaphase promoting complex. Means it allows the cell to move from metaphase to anaphase. Anyone remember what is the major function of APC? Degrading the cyclins. Yes or no? Degrading the cyclins, then and adding the phosphorylation to various molecules, that is securin. Remember? Correct na? Now, see here almost all the options are inappropriate option. Okay, see, you have to understand in this way. Originally, APC ultimate goal is, see, it is called as E3 ubiquitin ligase enzyme. It is only majorly responsible for destruction of the cyclins, never allow the cell to divide. It generally stops the cell division and it allows the cell to enter into the anaphase. Correct na? Which means what you have to understand, chromosome condensation and halt progression, this is a kind of true statement. These two we can definitely eliminate. It never directly or indirectly never involved in the protein synthesis, and it is not at all involved in the DNA repair. DNA repair mechanisms are stimulated at the G1 phase majorly. Now, according to the official answer key, they have given answer as the 1. Why they have given the answer as 1? Because metaphase is completed. Then we are entering into the anaphase followed by the telophase, which means we are at the end of cell cycle. Correct na? Once we are at the end of the cell cycle, the cell is going to enter again what phase? G1 phase, where it is going to prepare all the enzymes required for the DNA replication. See, technically we can consider this is the answer. Technically we can say. But you quickly recall what is required for the chromosome condensation. Anyone remember? What is that? Condensin protein. Very good. That condensin protein is activated by what? Very good. What is that? MPF. Mitosis promoting factor. You remember, they are made up of mitotic cyclins and mitotic CDKs. We read other CDKs named as Aurora, Polo-like kinases. Their function is what? Phosphorylating the condensin. By that chromosome condensation happen. But this is happening in which phase? Metaphase. But APC responsibility is what? To allow the cell into the anaphase followed by exiting of the cell cycle. Then it is entering into the G1. So indirectly APC is indirectly, I never use the word directly, directly it never involves. APC is indirectly involved in promoting the G1 phase. So it initiate replication and allow the progression of the cell cycle or allow the progression through the cell cycle, right? So according to the official answer key is 1, that is appropriate. Let's see, this is also 12th standard question, 11th standard question. First start. This is lac operon example, contains structural genes followed by operator, promoter, regulatory gene, and there is one more promoter for the regulatory gene. Promoter is the binding site for RNA polymerase. Operator is the binding site for regulatory protein. This is the switch which control whether the operand, whether the structural genes to be active or not. This is controlled by the operator, right? So operator, this is a common word, it acts as a switch controlling the access of RNA polymerase to the genes in the operand, correct na? If operator is free, which can move? RNA polymerase can move. So its movement can be controlled by the operator. It's like a switch. If we switch on, then only fan can be turned. Same way. If it is free, then only polymerase can move. That's why it can act like a switch. See, this is 10th standard CBSE question. Everyone commonly say that zygote is the area or the stage where synaptonemal complex will be formed, correct na? Which leads the formation of the tetrads. Okay, every student commonly know that it's a 11th standard NCRT question. See, the same lines also we read in the PPT. No, it's zygote, not packete. We read this question so many times. See, we discussed, right, transposons are scattered throughout the genome, correct na? It's a straightforward statement of the standard reference book. They are within exon interrupting coding sequences, there may be a possibility, but they never arranged. See, suppose this is a gene, this is a transposable element, it may inserted here, correct na? But the arrangement is completely different. Arrangement is what? They scattered or arranged throughout the genome, correct na? Some are confined to some areas, but in generally they are scattered. Some transposable elements are arranged in a form of a cluster. Even though they are arranged in a form of a cluster, they are moving or scattered throughout the genome. This statement is true, but it is not appropriate statement. Why? Because they are saying that confined to specific transposon cluster. Because on our DNA there is nothing called as transposon cluster. Wherever, because the insertion is a random insertion, wherever it wants to insert, it can insert on its own and it's scattered throughout the genome, right? The most appropriate or correct statement is option 3. So this is 10th standard question, correct na? So because vacuole allow the storage of various ions or the solutes which increases the turgor pressure. So simple question. See, there is, when we consider reason, friends, we should always understand this reason directly proportional to this assertion or not. That's what we have to do it. They are saying Okazaki fragments are formed during DNA synthesis. That is absolutely true. Correct na? When this Okazaki fragments are formed, whenever the DNA replication occur or whenever the daughter strand synthesis occur, Whether they are moving in the leading direction, whether they are moving in the lagging direction, that is secondary point. But the direction of the synthesis of the DNA always happens in 5 prime to 3 prime. Correct na? See, Okazaki fragments are also synthesized in which direction? 5 prime to 3 prime, but away from the replication fork. Correct na? So Okazaki fragments are formed during DNA synthesis, which generally happen in 5 prime to 3 prime. So answer is the 1. Correct na? You have to see reason is proportional to the assertion or justifying the assertion or not. They are asking false. Aram se. Anyone remember what is the major function of the Pol 5? See, it is generally involved in the SOS repair. It is coded by a operon named as a UMUDC operon. It is the name of the gene or name of the operon that can code for polymerase 5, which is generally involved in SOS repair. Correct na? But it do not have any proofreading activity. See why? Because this particular SOS repair, if you recall, we generally call it as what? Error prone repair. If they are having proofreading activity, error never happen, right? So this is the false statement. It belongs to the gamma family of DNA polymerase, catalyzed translation. Sometimes instead of using the word translation, they use the word bypass. Okay, bypass mechanism or bypass DNA synthesis or translation DNA synthesis. It catalyzes error prone. Since it is catalyzing the error prone, it do not have any proofreading. So answer is the 3. So E. coli DNA Pol 5 also called as UMD2C. It is the name of the operon that code for this enzyme. We call it as TLS polymerase, translation synthesis polymerase. This is also directly there in the slides only. Answer is right or wrong? Options are there or not? Which of the following enzyme is responsible for mitochondrial DNA replication? They are asking mitochondria. Anyway, polymerase alpha having primase activity, polymerase activity. Beta, DNA repair. Polymerase delta, one of the major DNA polymerase enzyme which is required for elongation. DNA polymerase lambda, bacteriophase. At the same time, it is also involved in the repair. Right? It is also involved in the repair. When it comes to the mitochondria, we are having a gamma. Right? So answer was not given. So simple. This is also 12th standard only. Twelfth standard it is. What's the answer? Which of the statements are correct about the spliceosomes? What we studied? Okay, answer first. They are asking spliceosomes. They are composed of RNA and protein. RNA and proteins together called as SNRs. Correct na? If only RNAs are present, we call SNRs. They recognize RNA sequence that signal for removal of intron. Ultimate goal of the spliceosomes are what? Remove the intron. What is the name of the mechanism? Transesterification. Is it? Removal of the intron. And they can produce different mRNA molecules by splicing it. What is that splicing called as? Alternate splicing. One mRNA, many proteins. Like, see, one mRNA with one mRNA give rise to what? Lag, JAD, YA. This cannot be the alternate splicing. Here, one mRNA giving rise to the different protein, like an antibody, right? Which is called what? Alternate splicing. What happened in the alternate splicing? Exons are skipped. Correct na? Exon 1, 2, 3, 4. 1, 2 and 4 will pair. 3 got skipped. Sometimes 1, 2, 3 got paired and 4 got skipped. This is called alternate splicing. So different proteins we get from one single mRNA. In mediate splicing at alternate site. This statement itself a wrong statement or incomplete statement. What is this mediate splicing at alternate site? No. So answer is what? 1, 2, 3 are the most appropriate and correct statements. This is also 11th standard, a 12th standard question. So, what is the homologous of Shine-Dalgarno sequence in the eukaryotes? What is that? Kozak sequence. How this can be recognized? With the help of tRNA, correct? With the help of until anticodon and codon pairs, this sequence will be continued. I mean to say, this scanning will be continued. But here, suppose this is the mRNA and this is the start codon. From the start codon, 10 nucleotide upstream, we are having a complementary sequence towards what? Towards the 16S rRNA present in the 30S. So this will come and pair here, is it? So which is binding of ribosomes to the mRNA during the initiation of translation. Binding of RNA polymerase to the gene, no. Binding of DNA polymerase, no. Binding of SNRPs or spliceosomes, no. It is only responsible for binding of ribosomes to the mRNA. They are asking processed mRNA. No, it's very simple. How does the mRNA look like? mRNA is generally coming from DNA, right? mRNA is generally coming from the DNA, which means whatever the sequences are present in the DNA, that also will come to the mRNA. But whenever the mRNA is synthesized in the eukaryotic organisms, it is immature. So since it is immature, what we are going to do? We are adding the cap, we are doing the polyadenylation and we are doing the splicing. Splicing means what we are doing? We are removing the intron. So they are saying there is one intron of 1000 base pair. Even though it is 10,000 base pair, we are going to remove it. If you quickly recall, this is mRNA. What we studied, entire mRNA never code for protein. Is it? What we can say, there is a coding region is present. We call transcription unit or a functional unit, we can call cistron. What is that? Cistron. And we all studied now at the 5' region what we are having? UTR, which is called as leader. And 3' also we are having UTR, which is called as trailer. Even though they are not coding for the protein, they are present in the RNA. They are not asking whether they are present in the RNA or not. They are asking length of final processed. Correct na? So what they contain here? Cistron generally contain codons, correct na? And stop codon. But here, suppose example, the coding region generally contain how many? 150. So 150 into 3, 450. Correct na? And UTR is 100, 550 and 3' UTR is 200. So how much it is? 750. Right? It's a simple one. Understand everyone correctly? Correct, correct. Everyone is correct. Which statement is false about the genetic control in prokaryotes? Slowly, slowly, slowly. You know, while explaining the slides, I use it to repeat this line 2 times, 3 times. Everyone online, what's the answer? Why? Because see, the moment you heard the name of gene expression, first main gene expression happens at transcription. Correct? Best example, you can consider heterochromatin and euchromatin. Generally, in the heterochromatin, gene is masked. As a result, what never happens? Transcription. mostly gene expression happens at the transcription at a lower level, at a translation, lower, it happens at lower level. But they are saying most of the gene expression are regulated at translation level, which is wrong statement. But RNA polymerase must bind to a promoter on the DNA to begin RNA synthesis. This statement is explaining about transcription. This question is about gene control or the gene expression. An active repressor protein inhibits RNA polymerase from binding to DNA. Is it true or false? What is the function of a repressor? Repressor will go and associate with what? Operator. Correct na? Then if operator is blocked, RNA polymerase never ever move. Correct na? Statement is right. Repressors, what we can say? Repressor control gene transcription by binding to operator. Statement is true. So the only false statement is option 2. It is so simple question. Which of the following mechanism is not involved in virus induced cell transformation? Let's take an example of bacteriophage. Remember what happens when the bacteriophage attacked on a bacteria first? Bacteriophage having its own DNA, that DNA is going to be released. And at the same time bacteria having its own DNA. Now generally what happened? This entire DNA of the bacteria is break down into pieces. Only this undergoes replication. Correct na? So what is happening here? So direct inhibition of viral replication never happens. Even in the transformation also, this is a transduction process. Here they are saying how this DNA is getting transferred. Whenever this transfer is happening, which of the following event is not involved? Insertional mutagenesis, what do you mean by that? This gene can introduce and cause the mutation. This gene can introduce in an area which can turn into oncogene. You remember in the cell signaling we discussed many of the viruses can act like a oncogene. Anyone remember? Spleen focus forming virus. Remember, whose structure is exactly look like certain growth factors. You remember, there are many growth factors look like a EPO. Correct na? So they generally activate the oncogenes. Inhibition of tumor, because if they are activating the oncogenes, definitely they are going to inhibit the tumor suppressor. So these are all the common processes or common mechanisms shown by the virus. During the time of transformation, direct inhibition of viral replication won't happen. Right? Answer is 4. This is again NCERT 11th standard question it is. Which is red light, which is far red light? Phytochrome is PR, is it? This is on PFR. So simple question. Which photoreceptor in plant is responsible for regulating flowering time? Flowering time. Now everyone, what is the answer? Is it A or B? Look into this properly. Check it everyone. Is it A or B? Everyone. Flowering time, which represents what? Photoperiodism. Everyone, online people, what is the answer? You forgot. These are the straightforward lines. Very good. Which is the phytochrome B. They are asking flowering time. Short day plants, long day plants, we have studied, right? Which is called photoperiodism. That photoperiodism is majorly shown or by the characteristic feature of phytochrome B, not the A. Okay? Please don't confuse. They are straightforward statements. If you answer a difficult question wrongly, there is no problem. If you answer simple question wrongly, there is no use of our preparation. See, this options, everything is wrong they have given. This question I believe last time they have given the marks, I strongly believe. All answers are wrong here. Options only wrong. See, they have given match the COVID-19 vaccines listed in column 1 with the correct platform they utilize for delivery of genetic material listed in column 2. Okay, so I want you to compulsory remember this without confusion here. And what we observed is even in the 2023, 2022 also we got the similar type of a question. So this time also there is a possibility of getting this kind of a questions. I want you to remember correctly. Which company is using what, is delivering what type of vaccines and delivering what type of vectors? Pfizer and Moderna company, right? They generally develop the vaccines which are mRNA based, right? AstraZeneca and J&J, Johnson & Johnson, they uses adenoviral vector vaccine. Anyone remember adenoviral vectors? In the last class of applied biology we discussed. Very good. What is that? Adenovirus only. Correct now? That is only we used as a vector. In case of plant vectors, what we used? TI plasmid and RI plasmid. So here we are using viral vectors. AstraZeneca and Johnson & Johnson, they uses adenoviral, right? Covaxin, anyway they have not given. It is also inactivated virus which is also a type of the mRNA only. Whereas Novavax, it is not RNA one, it is the protein. So what I want you to remember for the exam point of view, next time all the students remember. Pfizer and Moderna, these two companies, they are developing the vaccines which generally utilizes mRNA. This I want you to remember. Pfizer and Moderna. Whereas AstraZeneca and Johnson & Johnson, they uses viral vectors. So two companies are mRNA and two companies are viral vectors. So in that case, Pfizer and Moderna both uses mRNA only. Where, which one ma? Viral? Okay, okay, sorry. So Pfizer and Moderna is these two, correct na? Then AstraZeneca and Johnson & Johnson will be the viral vector. So options itself wrong. I want you to remember this correctly, okay? In the 2023 paper also similar type of question appeared, okay? Please remember them mandatorily. This is also a 12th standard simple question. See, we should always consider the in vitro condition and we have to understand. Everyone, online people, I think you read the what we can call rDNA technology in the ATPase Sir class. cDNA. What is cDNA? Complementary DNA library. Correct na? Here what we do it, and we read the genomic library also. Here what we do, we take all the, what we can say, all the vectors or all the genes and we will store it. So this is what? Final process generally. So anyway, first understand the given question. Which sequence of the following techniques and antibody engineering involving the use of transgenic mice engineered to produce human antibodies and subsequently immunized the target antigen to generate fully human monoclonal antibodies is correct. So what we are doing, see here, first we are using transgenic mice and we are producing antibodies, but these antibodies I cannot call it as a complete human monoclonal antibodies, because here most of the portion of the antibody prepared from mice, then this antibody which is prepared from the transgenic mouse I have completely converted into human monoclonal antibody, right? Nowadays whatever the antibodies we are using, completely human monoclonal antibodies only. In the last time we explained in the immunology why we don't use this mice derived antibodies, because they cause severe allergies. They are not compatible in the human, right? That's why we will completely use a humanized or human monoclonal antibodies only. So they are asking what is the sequence. First, whether it is a transgenic mice or whether it is a human, first we have to develop the antibodies. How we develop the antibodies? By hybridoma technology. Oh, like that also we can answer. Huh? Simple. Okay. This is, first one is hybridoma technology. The next one is combinatorial antibody libraries. What do you mean by that? We have prepared one antibody. Now by using a different type of genes, we will prepare set of antibodies and we will convert into the libraries, which means what? First, initially we have produced the antibodies which are coming from the transgenic mice. Later we modified into humanized. Correct na? Then we stored and we displayed it. Right? So this is the most appropriate. So first is production, making bulk copy, converted into the human use, and finally display. Simple, straightforward questions. Which of the following is not so simple? What's the answer? They are involved in transmitting the signals from extracellular ligands to intracellular. They consist of 7 transmembrane. They possess an extracellular ligand binding domain. They are found in plants also as well as in the animals also. They are not restricted to the prokaryotic organism. Answer is 4. What is the primary function of quorum sensing? It is also simple. Now what's the answer? Quorum sensing is a mechanism by which the bacteria start maintaining their density with respect to environmental condition. An example, the environmental condition is a stressful condition. If you quickly recall, the bacteria on their outer surface, it contains glycocalyx, is it? All glycocalyx of the bacteria come close together, they can form a rigid structure called as biofilm. Remember, so most of the immune cells cannot break down this particular biofilm. Now the question is, when this bacteria is making this biofilm, see here, understand the point. Does this particular biofilm is formed by single bacteria? No. Many bacteria are having glycocalyx on their cell wall. Many glycocalyx of many bacteria come close together, they can form the biofilm. By that they can protect themselves from the adverse stress conditions. That mechanism is called as quorum sensing. So first, bacteria receives or sense some chemicals such as oligopeptides. After sensing these chemicals, what these chemicals are explaining, the condition is stressful. Then immediately they will start synthesizing the biofilm in order to protect or in order to survive during adverse environmental condition. So coordination of population-wide behaviors in response to cell density. Quorum sensing is not the mechanism of gene expression. It never controls DNA replication. We don't use the quorum sensing for the production of antibiotics. A simple straightforward line in the PPT. What is the most commonly used tumor suppressor gene? P53. First identified is PRB. What's the function of every tumor suppressor gene? First they arrest cell cycle. Then they stimulate DNA repair. If the repair is not possible, they will allow the apoptosis. Correct na? Programmed cell death. So they promote, they never promote, they generally inhibit, They repair damage, they never repair. They are involved in the activation of DNA repair mechanism. You are getting the point, because P53 is a transcription factor which increases the transcription of those genes which are involved in the repair. They never involved in the, they never repair the damaged DNA. They activate those enzymes which are involved in the repair. Definitely they inhibit cell cycle progression and prevent tumor formation. They never ever induce the apoptosis in healthy cells. They induce the apoptosis in those cells where the DNA repair mechanisms are failed. Right? Simple. Answer is, what is that? 3. TLR receptors, very common. It's okay, remember, TLRs are not at all GPCRs, okay, and they resemble, I am using the word resemble, they resemble like enzyme linked receptors, but they are not enzyme linked receptors also. In simple words, they are not GPCR, they are not enzyme link. If it is an enzyme link, what we studied, they are having extracellular domain, ligand, transmembrane domain and cytosolic domain or intracellular domain. Most important is what? Which is having kinase activity. When it comes to the toll-like receptor, they contain extracellular domain, intracellular domain, but they do not have intrinsic kinase activity. Hence, they are not RTKs. Even they are not GPCR, because they will transpan hardly one time. They are not transpanning 7 times like GPCR. The only thing is that they are having extracellular ligand binding domain. Right? Even though intracellular is there, it do not have the kinase activity, so it will be completely inactive. Mostly they are having extracellular. So it cannot be a 7 transmembrane, it is having extracellular ligand binding domain. It do not have kinase activity. Nuclear localization signal function is what? Allowing the protein to enter within the nucleus. That is associated with the proteins. It is not associated with the receptor, right? Remember, it is having extracellular ligand binding domain. And they are single pass. See, these are all just from the slides. B1. Ma? Answer 1. Na? That much fast, how you did? What is the answer? Wingless segment polarity gene. What is the example of wingless, and we read one more gene? Very good. What is that? Aram se, slowly. Wingless and engrade. You remember? We have drawn the diagram and we read. Wingless uses what kind of receptor? Anyone remember? Wingless uses frizzled and a TLR receptor. Engrailed uses? What is that? Receptors I am asking. Very good. Hedgehog. One is the ligand and one is the receptor. Correct na? Remember everyone. Anyway, wingless and engrailed is the segment polarity. And what about the hedgehog? Of course. Then what is the other gene? Decapentalpagic gene, which is dorsal ventral. Bicoid and hunchback. There are anterior posterior axis formation. Right? You can remember, those are all just examples, straightforward. Correct. Patched receptors, right. Frizzled and patched. Patched and smooth end receptors. It was so simple, we can answer this. They are asking a question, what are the correct sequences of apoptosis? In the apoptosis, finally what happens? You have to remember. In the apoptosis, cell death will happen. What are the enzymes are finally involved in the apoptosis? Caspases. Correct now? With that also you can answer easily. right? Because caspases are directly destruct the cell by creating a pores, correct na? Now, first, what is the function of tumor necrosis factor? You have to remember this is the growth factor. You are getting the point. And TNF are also attached to the receptors first. You are getting? Because without receptor activation, there won't be any activation of what? Tumor suppressor genes as well as the apoptotic proteins. You have to majorly focus on which are pro-apoptotic proteins and which are apoptotic proteins. These two, it is very much important, we all has to remember for the exam. So first, what is happening? Tumor necrosis factor acting as a growth factor will come and activate the receptor. Upon receptor activation, there is activation of what? P53. Then P53 is going to activate the apoptotic protein. Apoptotic protein is going to stimulate the caspases. Caspases start the process of apoptosis. Okay, this is also simple, straightforward only. This is also straightforward. Every question is so simple question. Actually, it was not given correctly in the C. elegans. I don't know. See, the question is about C. elegans. All the students remember C. elegans generally follows MAP kinase pathway. We have other name here. The name of the growth factor LIN2 and LET23. Right. See, in more simple words, an example I will tell you. Suppose I am taking MAP kinase pathway. Example. What is the major growth factor that using the MAP kinase pathway, which is called as EGF? Epidermal growth factor is the name of the growth factor, uses the MAP kinase pathway. Finally, they will activate variety of kinases and finally they show the growth. And they are asking about the C. elegans. In the C. elegans are also same MAP kinase pathway only will happen. So, this is the name of the pathway. The MAP kinase pathway having one more name named as LET23 pathway. That's all. Right. So, all options are wrong options. For this also definitely they have to give the answer. Right. It's not a vint, it's not a notch, it's not a hedgehog, it's not a TGF pathway also. Right. Please remember that. This also I explained on the first day in the previous classes, not in the revision. In the revision, not in the revision, before that I have explained it. The major growth factor required. Slowly. They are asking proximal distance. This is the first line in the slide only. Please remember, in the proximal distal axis of the limb, always there is an involvement of FGF, fibroblast growth factor. Okay. After fibroblast growth factor, there is an involvement of sonic hedgehog. There is no Wnt beta catenin and there is no TGF pathway also. Please remember, this is there in the slide itself. Slide of this developmental biology. This is straightforward again. Stem cells. Everyone. See, don't think that you have to answer all 100 questions. I told you, right, you have to focus on 75 questions correctly. Okay. No need to, discourage. Definitely from the blastocyst. That is a straightforward question. Simple. This also we discussed 2 to 3 times. Slowly. You are not reading even. First, what is the primary organizing center? Organizer is the one which, what is organizer here? Very good. Dorsal lip of the blastopore. Act like a organizer. This organizer is controlled by Newcomb center. Major organizer is what? Dorsal lip of the blastopore. And what is the name of the chemical that is majorly required for the control of this Newcomb center? Which is beta catenin. Correct na? Which is beta catenin. It's a so simple question. They're asking, no, the primary organizing center. It's okay, nothing like that. So this is also we read. So simple question. Every one of you. So simple. All of you. Which cell surface receptor is responsible for sperm egg binding in the process of fertilization? Integrin is not the cell surface receptor. Integrin, cadherin, selectin, these three are what? CAM molecule. There is only one receptor on the zona pellucida. We have ZP1, ZP2 and ZP3. So they have given ZP3 receptor. C4 and CAM pathways are more efficient than C3 pathway. Of course true. Correct na? How C4 plant is more effective? Because of what anatomy? Kranz anatomy. Why CAM pathway is more effective than the C3 pathway? It shows temporal separation. That is closing of the stomata during daytime and opening the stomata during night time. So C4 plants are spatially separated, whereas CAM plants are temporally separate. That's the reason they can avoid the photorespiration and fix the carbon very efficiently than C3. So assertion is true. Reason is proportional to the assertion. So simple it is. Slow. So anyway, we can easily analyze the question. Symplastic, it is always associated with the plasmodesmata. So where is plasmodesmata? Okay. So it is definitely option 2. Correct na? Active, see we discussed it, right? Phloem loading and unloading. Both are mediated by apoplastic and symplastic. Both. Right. Now they are asking about active unloading, passive unloading and apoplastic loading. So apoplastic loading means what? Means what? Sucrose is getting loaded by using apoplastic. You remember what is apoplastic loading and symplastic loading? That time we discussed. Forgot. Phloem loading. Phloem loading via apoplastic and symplastic. It depends on what cells? Depends on companion cell. Remember or not? We have different type of a companion cell. Transfer companion cell and intermediary companion cell. Depending on the type of the companion cell, the type of transfer will happen. Tell me, what is this apoplastic loading means? Apoplastic, what we discussed, it always move across the cell membrane and intercellular space. Cell wall and intercellular space. Correct na? So apoplastic loading, sucrose is transported across the cell membrane into the phloem via membrane transporter involving the apoplast. Absolutely true. Correct na? Now what is the B now? Active is energy dependent process. Sugars from the phloem into the sink often again is concentration. And what is left out? Passive unloading. Correct na? Without using of energy. From higher to lower or along the concentration gradient. So what's the correct answer? Option 2. Right. This is all informative only. What are the three pigments we have studied? Phytochrome, cryptochrome, phototropin. Phytochromes are which light? Red light. Cryptochrome and phototropins are blue light. Correct, na? So remember, cryptochromes are found both in the plants, highest in the plants, little concentration they are also found in the animals. They are also required for the variety of processes including the flowering. Whereas when we consider the phototropins, they are also blue light pigments, but they are only plant restricted. They won't be found in the animals. Cryptochromes are majorly involved in the phototropism, while phototropins regulate the circadian rhythm, which is opposite. Phototropins are the one which are responsible for photoperiodism and the chloroplast movement. So statement is opposite. Cryptochromes are blue light photoreceptor found in plants and animals. Phototropins are specifically found in plants, right? Phototropins are responsible for the opening and closing of the stomata in response to blue light, whereas cryptochromes regulate the chloroplast movement. Opposite. Correct, na? Phototropins are the one which are responsible for the opening and closing of the stomata also, and they are only required for the movement of chloroplast. Both cryptochromes and phototropin function as a blue light photoreceptor in plants. This statement is true, but cryptochromes are more involved in photomorphogenesis and phototropins are induced gene expression. Which is involved in photomorphogenesis? Phytochromes. Phytochromes involved in the photomorphogenesis. Cryptochrome and phototropins are involved in the photoperiodism. Cryptochromes are involved in the flowering. So simple, you can remember 1 points. Okay, this statement is also wrong. So simple statement, phototropins and cryptochromes both are blue light photoreceptor. Cryptochromes are exclusively found highest in plants but also in animals, which is responsible for flowering. When it comes to the phototropin specifically found in the plants required for chloroplast movement, stomatal opening and photoperiodism. Right? Answer will be the 2. Everyone, online people, please give the answer. This is also so simple question about the terpene synthesis which we read. This is kind of twisted. Shafia, cryptochrome and photochrome, that is true, no? But phototropins are more involved in the chloroplast movement. Cryptochromes are not at all involved in the photomorphogenesis. Is it? Photomorphogenesis is mediated by phytochromes. And what is the answer for this? Second, huh? Okay. Others? Slowly analyze. First, what is the terpene biosynthesis we all discussed? We have 2 pathways, mevalonoic acid, MEP pathway. By using mevalonoic acid pathway, we synthesized IPP and DMAP. Correct? Na? This is a 5 carbon, this is a 5 carbon. So definitely first we have to synthesize the precursor, because active terpene is generally made up of 10 carbon. These are all hemiterpenes which are 5 carbon. So first we have to synthesize the precursor. Formation of IPP and dimethyl aerophosphate. Correct now? This is true. So these, which one we can eliminate? This one we can eliminate. Then conversion of IPP and DMAPP into various terpene catalyzed by terpene, because this is a 5 carbon and this is a 5 carbon. Both 5 carbons are combined and give rise to one. What terpene? 10 carbons. 10 carbons are called as? What is the name of the 10 carbon? Monoterpene. Correct, na? So definitely option 2 is right. Now, after monoterpenes, what we synthesize? Sesqui. Then followed by di. Right. So after IPP and DMAPP, these two combined give rise to, followed by sesqui. And so on, we are going to synthesize large terpene. So exclude synthesis of terpenes in the cytoplasm of plant cell through mevalonate pathway. Okay. Actually, this is outer statement. The question is about the synthesis of the terpenes. Definitely first we have to synthesize precursors. After that both precursors has to be added, giving rise to the monoterpene, sesquiterpene and so on. But they are adding a statement saying that exclusive synthesis of terpenes are happening in the cytoplasm of plant cell. The statement is true, okay, but this is not in the sequence, because after the precursors we have to synthesize monoterpene, sesqui, and so till here the statement is over. The additional statement or outer statement is about their location, which is the 3. Answer is option 3. Do not confuse with that. This is also 10th standard question, CBSE question. Primary form. Primary form of nitrogen assimilation in the plants. Before the assimilation, what we do? Reduction. Correct or not? Correct or not, ma? So first we fix the nitrogen in the form of NH3, correct na? Or the NH4. Then what we do? We will produce NO3, then followed by NO2, correct na? Then assimilation happens, correct na? Answer is what? Nitrate reduction to the ammonia. Simple. It is also a common question. Which of the following is a common chemical defense mechanism used by plants? Whenever a pathogen infects the plant definitely going to release what? Certain secondary metabolites, correct na? Which shows antimicrobial property. What are those? Phytolexins. We read now the same chemical also. So is there any necessity to activate the guard cell when the pathogen attack? There is no need to produce the cell wall degrading enzymes. We no need to increase the water uptake. Plant has to protect itself against the pathogens. So it is going to release certain chemicals which are having antimicrobial property, which are phytolexins. So simple. We already, the same question was repeated in the matching previously. Plants to the heat stress. What's the answer? Production of osmoprotect like proline, betaine, trehalose. Correct na? Antioxidant synthesis has to be increased. There is no role of stomata here and there is no role of leaf senescence here. Correct na? So first is definitely P680 followed by pheophytin followed by plaster. Since the pheophytin was not given, plastoquinone will be the answer. Simple. Don't confuse, okay? Don't confuse. So simple question it is. What is the answer? 3, yeah. How come 3, man? See, the question is, when a person runs at a velocity of this much speed and comes to a rest after 30 minutes, then which one of the following is the correct statement? Now, whenever we are running, we spend more oxygen or less oxygen? Definitely more oxygen we require. So whenever we are running fast, we require more glucose or less glucose? So generally glucose breakdowns, correct na? Giving rise to what? Carbon dioxide and H2. Whenever we are running continuously, glucose got breakdown, oxygen level is also falling down. Then what happens? This glucose gives rise to pyruvate. Pyruvate convert into lactic acid, get accumulated. Correct or not? Lactic acid never get breakdown here. Correct na? So which of the following is, see, this is true, I agree. Lactose in the muscle oxidizes into carbon dioxide and H2O. Lactose never give rise to the carbon dioxide and water. Lactose in the rest condition converted into pyruvate by a cycle called as Cori cycle. Correct na? That is, lactic acid is getting converted, it never breaks down. Lactic acid accumulation happens generally, right? So the glucose in the body oxidizes into carbon dioxide and water. Simple, don't confuse. This is straightforward question which we read. IRV, ERV, so many times I explained this. How much tidal volume? 500 ml. Very good. Correct na? What is alveolar ventilation means? Means how much is moving to the alveoli. How many breaths will happen? 12. Correct na? 12 into 350 is how much? 4,200. So if you consider total 12 into 500, this is 6,000. This is total, but we should always consider the how much of the amount of the air is reaching into the alveolus. And what is the question? See, generally when we are taking a normal breathing, 500 ml will enter, normal breathing. But what they are saying, the maximum of volume of air expired from the lungs by a person after taking a maximum, what does it mean? They have taken deep inhalation and they have done deep exhalation. Then what happened in this condition? More amount of the air is going out. Yes or no? So originally this is the tidal volume. This is normal. I don't know whether you remember or not. What is called IRV? Inspiratory reserve volume. Forcefully how much you are taking inside by forceful inhalation. What is ERV? Expiratory reserve volume. They are asking that only. Expiratory reserve volume. It will be varied from person to person. Okay. Maximal expiration after maximal inspiration is called as vital capacity. So vital capacity can be calculated by IRV plus TV plus ERV. Right. So tidal volume is fixed, which is 500 ml, and this one what we can call the IRV. So it is 3000 to 3500 again person to person. This is 1000 to 2000 again will be changed from person to person, or sometimes 1500, 1200 changing person to person. Okay. What is the answer given here? So you have to remember they are asking about the ERV and VC, vital capacity. This is 11th standard anatomy question it is. They are asking incorrect. You can know what is the answer? Why? The auriculoventricular walls are connected to the papillary muscles by chordae tendineae. Correct na? Papillary muscles are chordae tendineae. Right? What is bundle of hairs? They are the network of branches which are responsible for carrying the impulse. Right? This is definitely wrong option. Interauricular septum has a small oval patch called as fossa ovalis. Right? The right auriculoventricular apparatus is guarded by tricuspid, and one side we will be having a bicuspid or mitral wall. So simple. Return. Return is what? Generally from where to where it is happening? From the tubule into the blood, which is selective reabsorption. It's not a secretion. So simple, straightforward. Everyone, online people, be active and answer the questions, right or wrong. Sriyagna, answer. Slow, ma, slow. According to the accepted concept of hormone action, if receptor molecules are removed from the target organs, we studied the properties of hormones in the first class. Hormones are low molecular weight or some are high molecular weight, and we studied that hormones are always released at the lower concentration. Lower the concentration, higher the activity. And we studied site of production, site of action is different. They always carry blood as their stream of transport. And if they want to work, definitely they require a receptor. Whether it's an intracellular or extracellular receptor, they require the receptor. What we have done? We have removed the receptor. So without receptor, hormone never show any action. So the target organs never ever respond. The target organs never continue to respond, will continue to respond to the hormone but will require higher concentration. Wrong. Because we have removed the receptors only. The target organs will not respond to the hormone. Without receptor, where the hormone attaches, without attaching the hormone, organs never works. Simple, straightforward. So simple. Test tube baby, in vitro or in vivo? In vitro, that's all. IVF. So simple, quickly. So it's a simple definition. See, what generally we do when there is a problem with the men or women, we collect the sperm, we collect the egg, we will keep it in the culture plate, where we provide all the necessary nutrient mediums. Then what we will do? We will allow the sperm to become capsitated, sperm become matured, sperm fuse with the egg, and then there is a formation of the zygote. That we are going to implant in the mother's uterus. Rest of the process is absolutely normal. Fertilization of the egg and growth of the embryo are affected in a large test tube. A premature baby is reared. Fertilization of the egg is affected in the female genital tract. No. Right? They are asking statement regarding the test tube baby, which is in vitro fertilization. Myopia, we generally consider, what we can say, short eyesight. Okay. Cyclopia, single eye. Okay, that is called single eye. So remember this, it is not normal. Okay, the diameter of the eyeball is longer than the usual, causing the short, near image to become blurred. Okay, you have to remember the characteristic feature here. Okay, it is not short, okay, or short sight we can say, but eyeball is longer than the usual. Please remember that. This we already read this. Okay, I think I have solved this question in the middle. This is from where? Extension of the Mendel principles. We read now a mechanism named as epistasis. What are the type of epistasis we read? Dominant, duplicate dominant, recessive, duplicate recessive. Very good. Now, what is the question? Dominant LLP produces a pigment called as yellow. Clear? Recessive LLP never produce any pigment. Very clear. What is the function of dominant LL? Whatever the pigment produced by the dominant LLP, that pigment can be converted into orange. You're getting the point, by Q. You're getting the point. What is the function of dominant P? Producing a yellow pigment. That yellow pigment is converted into by Q. Correct, na? Now can I say one point here? If I want to get the orange, how many dominant alleles are required? Definitely I require 2 dominant allele, capital P and capital Q. This is clear. Small p produces no pigment. Next, small q does not affect the pigment. It is clear up to here. Now, what is the question? The recessive allele Q, okay. A plant with the genotype PPQQ has orange flowers. Correct or not? Why? Both dominants are present. A plant with genotype PPQQ, because not even single dominant, but we generally require 2 dominants, has white flowers. If 2 heterozygous plants PPQQ are crossed, what is the generally we get? We have 2 dominants. Correct or not? Result also they only given 9:3:4. So definitely it is what epistasis? Recessive epistasis. So when 2 heterozygous plants are crossed, the expected phenotypic ratio is 9:3:4. You have to remember that. The interaction between the P and Q genes result in the dominant QLL converting the yellow pigment produced by the dominant PLL into, while the recessive PLL produces no pigment. Correct or not? Of course it is true. Okay. It's a simple question only, but it takes time to understand. Others, everyone online? You. Quick. So simple, I don't know what is this question. Everyone, what is the answer? What happened? They have taken 2 strains. One is F plus. One is HFR and another one is F minus. HFR having, is it auxotropic or prototropic? All are prototropic. Prototropic means what? Plus. Means having the ability or capability to synthesize everything. Correct na? Then what is, so F minus strain is what? Recipient. All are auxotropic. Now the mating has happened, the mating was interrupted every 5 minutes. And they are asking what is the order of the gene sequence. Definitely first leucine will move, threonine, lactose, galactose and Aram se read it. What's the answer? Which type of chromosome translocation involves the exchange of genetic material between non-homologous chromosomes without any net gain or loss? Correct na? Which is reciprocal translocation. Simple. What is the name of the repair mechanism that is involved in the repairing non-homologous, in the non-homologous recombination? I mean to say, double strand breakage repair can occur by using non-homologous recombination. What is this process called as? We already read mismatch repair is responsible for repairing the mismatched bases. Base excision repair is responsible for whenever base got mismatched. You remember, best example, whenever a process of deamination occurred, we get unnatural bases, xanthan, hypoxanthin, uracil, that can be repaired by base excision repair. Of course, we use non-homologous end joining is the major repair mechanism which is involved in double strand breakage repair by using Non-homologous recombination method. Non-homologous recombination means what? Recombination is happening between two non-sister, we can say, which are non-homologous. It cannot be the homologous. This is little tricky question. This is very good question. This is really good question. See, originally in a cross between variegated leaf plant female and a green leaf plant male. See, in the question, what they have not mentioned is maternal inheritance. Correct, na? In the question, they have not mentioned, or they have not mentioned mitochondrial inheritance. Correct, na? Originally, in a cross between a variegated leaf plant female, which contain both chloroplast and leucoplast, and a green leaf plant male, what will be the expected phenotype of the offspring with regard to the leaf color? If it is really maternal inheritance, definitely we get a variegated leaf. Correct, na? Definitely. If it is. If it is not, we have to get green. Correct, na? If it is not, means if it is not a mitochondrial inheritance, because compared to the variegated, green are a kind of dominant, right? But they have not said whether it is following the maternal inheritance or not following the maternal inheritance. So option 2 and 4 both are correct, but the most appropriate will be the 4. Offspring leaf color cannot be predicted due to the random segregation of chloroplast. Why? Because here chloroplast is present in the variegated and leucoplast. Here also chloroplast. Correct, na? So we cannot justify, whether it will be a green or whether it will be a variegated. So the correct answer will be the 4. Don't confuse, okay? Everyone online, people? In a study of a height in a particular human population, the heritability h square of the height was found to be 0.8. Which of the following statement is most accurate? What is the difference between option 1? So these two we can eliminate easily because it is definitely 80%. Now what is the difference between option 1 and 2? 80% of an individual's height is determined by their gene. So due to the variation in the genes, the height difference will occur. So 80% of the variation in the height in this population due to genetic difference, or due to the gene variation, there is a variation in the height also. A more simple words, more the variation in the genotype, more the variations in the phenotype. That's the point. So this is a 10th standard question again. They are asking, friends, accurately. Slow, slow, slow. How come first? Now what's the answer? They are multicellular organism. Bacteria is what? Prokaryotes. They do not have nucleus. They never possess peptidoglycan. They typically possess pseudopeptidoglycan. Is it? Archaebacteria, or we can call it S layer, slimy layer. Correct, na? So this cannot accurately describe. They are unicellular organisms with the prokaryotic cells, right? Accurately described. These are all informative only. I don't know what is this question and why they have given. They are asking not. which is not. We generally know that, what is the definition of a cell? See, cell is the structural and functional unit of the body. Group of cells belonging to the same size, same shape and same function form tissue. Group of tissue organ, group of organs, organ system and body. Tissues are made up of cells that have similar function, origin and specific function, right? Each organ in a plant or animal body is composed of a single type of tissue. No, right? Each organ in a plant or animal body is composed of many type of tissues. Organ systems are collection of organs. The respiratory, false, correct only. The respiratory system is a network of organ, tissues and structures that deliver oxygens to the cell and remove carbon dioxide. Answer is 2. This is also 11th standard. We read now that the same definition. What is called phylogenetic relationship? Why we read this? To understand the relationship between. They are asking which of the following statement about a phylogeny or evolutionary tree is correct. What it is? Why we use the phylogenetic tree? Phylogenetic tree is generally explaining the evolutionary relationship among the organisms in a taxa. Correct, na? The tip of the tree represents the common ancestor of the taxa. That is correct. But that is not explaining about the phylogeny. The nodes on the tree represents group of descendant taxa. That is also true. A phylogeny represents evolutionary relationship. Phylogeny do not provide information about it will provide the information about the common ancestor. These are all again informative. See, this is informative. I want you to remember tropical rainforest, deciduous forest, deserts and mangroves. Please remember this. Okay. Sometimes they don't give the word mangroves. That time deserts comes last. Okay. In that way we can remember. You see, that only botany people can answer. This is informative, but I want you to remember there is nothing to explain here. Myristica swamps are rare and significant. They found majorly in the Western Ghats. We can't find exactly Myristica swamp. Similar to the Myristica swamp is also found in the Southeast Asia. Now you remember correctly, okay? It's just an informative question. This is also informative. This is for zoology. Roundworms can grow up to 20 centimeter long and spread from infected soil or feces? No. Only tapeworms can grow up to 20 centimeters. Pinworms are a type of flatworms. We can take nematodes, right? That lives in the intestine and spreads by laying eggs in the host body. Remember this. Hookworms are intestinal parasites. No, one second. It is wrong, right? Oh, they are not flatworms. No, they are roundworms. I'm so sorry. Right. They are roundworms. Absolutely right. Right. Correct. Tapeworms are flatworms that live in the intestine and spread by laying their eggs in the host body. Right. Correct. Hookworms are intestinal parasites that attaches to the host intestinal walls and cause anemia. 1 second. Pinworms, a type of the answer should be 2, right? Yeah. Pinworms are nematodes, not flatworms. Right. Hookworms can cause the anemia, right? Tapeworms are flatworms. Roundworms can be 20 centimeter. I forgot. This though we cannot remember. You all has to remember correctly. Okay. So pinworms are what kind of worms? Okay. Pinworms are roundworms. So please remember the statements. Anyway, this is common. They are asking not. Which of the following is not considered a major driver of the bio? When the biodiversity can be changed due to the habit destruction, climate change, introduction of an alien species or invasive species, genetic diversity or species diversity or ecosystem diversity never changes the biodiversity. We know, right, commonly that one. Why the character displacement will occur? To avoid the competition, which means we are minimizing our niche, right? Correct or not? Increasing the genetic drift, enhance the reproductive success, minimizing the niche overlap. It never promotes interspecies hybridization. It's a simple question. Levin's modalities. See, you can easily answer what is called stability. See, stability means, friends, in a simple words, I can simply say like this. Rate of immigration should be equal to the rate of emigration. Right? Which means a balance between the local extinction and recolonization rates. Right? So that is what generally called stability. This is proposed by Levin's. It's a straightforward question. Interspecific. Inter means what? Within. This is intra, between. Answer slowly. Sorry. Right. Intra, within, inter, between. Right. Which of the following scenario best exemplify interspecific competition, and why does it not represent intraspecific? Everyone. Everyone, what is the answer? Why second one? No, what is the second one generally representing? Intra or inter? First understand the given question. Which of the following scenarios best exemplify interspecific competition, and why does not represent intraspecific competition? Tell me. What is the first condition? Two male lions in a pride compete for dominance and mating rights within the same group. Now, forget about this. Tell me, everyone, what is the first one? It is inter or intra? Next one? Why? Because there are different species are there. Third one? A flock of sparrows which are belonging to the same group. Several individuals of the same deer species. Correct, na? So the answer will be the option 2. Correct. Slowly. This is also same simple question. Echo, tone and edges. See, ecotones are the transition zones or also called as buffer. See, between two zones, so this is a common zone. This is what we can call it as the ecotone, right? So, ecotone act as a buffer zone that can protect a nearby ecosystem by trapping pollutants and preventing their spread. Ecotones are areas where competition between species is minimized. No, ecotones provide consistent, no one can provide the climate condition. Ecotones are not at all marine environments. Okay, these are all factual statements which we have to remember. Straight lines. What is the question? Primary succession, secondary. How can we remember? With soil, without soil. With the primary substratum, without primary substratum. Others? Everyone give the answers. We read now good example. What is the example we studied? Glaciers. Glaciers are what? Completely occupied by ice. After volcano or after lava, it is converted into bare rock. Then which species comes into the picture? Lichens. Correct now? Which is what succession? Primary succession. A forest regenerating after a fire. This is what? Secondary. So we can eliminate. The formation of a new island from a volcanic activity represents the? It is representing what? Primary. A newly exposed glacial landscape developing a community of plants represents primary because it starts with a bare substrate and no prior biological influence. An abandoned agriculture field means soil is already present. So it is definitely what succession? Second. Answer is 3. Good. This is the only question that we haven't discussed in the evolution. No, not this one, not this one. Not this one. This you can explain with the common sense. Which of the following statements best explain the interconnected impact of climate change on biodiversity loss and why it is crucial to understand this relationship for the policy making? So understand the given question very correctly. Which of the following best explaining the impact of climate change on biodiversity loss? You have to understand if there is a change in the biodiversity, definitely which leads to the climate change. So if we want to monitor this climate changes or if we want to keep the climate changes normal, first we have to stop the biodiversity loss. For that reason we have to make the policies. So first we have to understand why this biodiversity loss occur. Now we discuss now, suppose this is a forest. Why in case of a forest, why the climate changes generally occur? Because of the biodiversity loss. Why biodiversity loss occur? Due to habitat destruction. Correct or not? If I remove the entire habitat, if I clear for the purpose of agriculture, what happened to those species? Those species become completely extinct. Entire biodiversity will be lost. Human activities such as burning fossil fuels and altering land cover contribute to climate change, which in turn exacerbate biodiversity loss by altering habitats and ecosystem. It's very simple. It's a this is what do we call anthropogenic activity or human interference. Okay, remember this. They are simple questions. We already read now. They are asking friends oldest to recent. Others, everyone? Which is what? Cenozoic, Meso, paleo, proteo as well as the archaeozoic. So simple question. Natural selection, Darwin's theory. This is little twisted, that's all. Most of the people get confused here. Find out which is last. Survival of the fittest, variation and their inheritance, then correct na once correct then struggle for the existence, no natural selection, struggle for the existence, variation and their inheritance, this is definitely true, survival of the fittest and natural selection. The same characteristics will pass. So what is the option correctly here? 1, which means 4, 2, 3, 1. Is there any option? Right. Simple. Analogous. See, these are all 11th standard questions. We shouldn't take this much of time. But same function, origin is different. Everyone. Which of the following pair represents analogous? Others? Which we can eliminate? 1 and 4 we can eliminate. Then? Everyone's. 4 limbs of horse and hands of human beings. What do you have to and what about the third one? Oars means, which they use it to move, like a fins. Which is most appropriate? See, both are more appropriate only, okay? See, the answer key they have given 2 or 3. Okay, both are correct only. Slowly. They are homologous, na? See here. You remember I told you homologous is divergent evolution, analogous is convergent evolution. So they are all homologous, which follows divergent evolution. Remember, there is nothing to explain over there. We no need to use our brain. This is also 10th standard question. The present giraffe has a longer neck compared to the ancestors because of, which is the natural selection. Right? Simple question. And what is this theory? Lamarck theory. Okay. Same thing we read na, same human evolution, Homo sapiens. First we read, you remember, Australopithecines, then Africanus, then Erectus, Neanderthal, Cro-Magnon, nothing but Homo sapiens. You are remembering? Homo sapien only, Cro-Magnon, and modern man. Correct, Shafia, correct. They are homologous, right. They are homologous. 4 limbs of horse and human beings, right, they are homologous, right. So answer is 3. Right, correct. Sorry. What is this? Okay. What it is? It is called as what? Latent learning. Or sometimes they use the word mimicry. Okay, that also a correct statement. Right? Simple. See, there is no great concept to explain over there. It's not like a NET exam. This is a good question now. At least if you remember one also enough. Glutamic acid, it is from the Corynebacterium. Easily we can answer the question. And polymyxin, Bacillus polymixa. So even though you know, we can finalize these two and see glutamic acid, which is the Corynebacterium. So answer will be the option 2. Okay. I want you to remember them correctly. Remember, focus on remembering. Glutamic acid, Corynebacterium, glutamycin, vitamin B12, Propionibacterium, dextran, leuconostoc. Dextran is a sugar derivative, right? Polymyxin B from the Bacillus polymixa bacterium. See, it is called repeated back crossing. Oh, I told you right. Others? Remember in a simple words, introgression, which is equal to the hybridization process. And this hybridization, when it occurs frequently by using back cross method. In more simple words, the continuous or repeated hybridization by using back cross method, which is called as introgression. Okay, remember this. Repeated hybridization with back crossing. That no subject is required, simple question only. Starch is not the part of the growth medium for animal cell culture. Animal cell do not require the starch, this is true. And a normal cell need no external factors for its survival. It requires, right? It is anchorage independent, it is anchorage dependent, right? Only cancer cells are anchorage independent. So this is a false statement. So one is true, one is false. Biosensor, this is the question from unit 12. Okay, I want you to remember the first event is entrapment of enzymes, then cross linking, enzymatic reaction and response. Nothing will happen if you don't answer all the questions. I told you, right? You no need to necessary to answer all the questions. Out of 100, minimum 75 to 80. This is again good question. See, remember, excluders are always associated with the plant root. Suppose example, this is a plant root. So from the soil, lot of toxic substances will enter into the root. From the root, they are going to enter into the plant. Excluders are those which restrict the entry of the toxic metabolites into the root hair. So they will trap them in the root level itself and they will try to eliminate them. So they can restrict heavy metal ions to the roots and detoxify them, right? So then heavy metal protein transporters, which are responsible for a process of transgenesis. Hyperaccumulators. See, it's very simple. Hyperaccumulators are those type of agents. See, here you remember, excluders are never ever allowing them to enter into the root. Even though they enter into the root, they detoxify them and they will allow them to go out. Whereas hyperaccumulators, what they will do, they may come up to the shoot level, they may come up to the top branches also. From there they can be eliminated, which are called as hyperaccumulator. Means one can eliminate the toxic materials from the root level, one can eliminate the toxic material from upper branches and shoot. Right. Then what is left out? High biomass non-accumulators. Example are Salix species and Populus. Where we read this, to anyone remember? Salix, Populus. We read something called as, what is that? Hydrozier succession. Remember, we read different stages of the hydrozier. Ulnus, Populus, okay, in the woodland stage, right? This is very good question which you have to remember along with the examples. That's okay, fine. If you don't know, we have to leave it. They are asking incorrect. Recently also we discussed this. Recently also we discussed in the last classes. In the last class I explained this only. It's so simple, right? We have Ti plasmid, we have Ri. This causes tumor. Tumor-induced plasmid, this is what? Root-induced. Agrobacterium tumefaciens is responsible for what? Root tumor. And Ri, this is for what? This is for shoot tumor and this is for the root. They are saying Agrobacterium tumefaciens is responsible for hairy root. No, Agrobacterium rhizogenes is responsible for hairy root. Crown gall disease depending on the presence of Ti plasmid. In somatic embryogenesis, embryo initiation needs a high concentration of 2,4. What is this? 2,4 dichlorophenyl acetic acid. What is this? Synthetic oxygen. 2,4 dichlorophenoxyacettic acid is the most commonly used chemical analog of IAA. IAA is a natural oxygen and this will be what? Synthetic. You remember or not? 2,4 dichlorophenoxyacettic acid, 2,4,5 trichlorophenoxyacettic acid. Remember, these two are said to be what? Synthetic oxygen, which is analogous to the natural oxygen, IAA and IBA. This is 12th standard. Exactly, this is actually the same question repeated in the NEET also. What is the answer? Insect resistant is what? BT gene. BT cotton, BT brinjal, right? Golden rice contain a precursor named as beta carotene. It is the precursor for which vitamin? Vitamin A, which if the deficiency of the vitamin A leads to the giraffe thalmia. That's why they induce the beta carotene gene into the rice. Then rice starts producing beta carotene, so automatically there is a production of vitamin A occur. Correct na? So upon injection of the beta carotene, the rice color shifted to the light gold. That's why we called golden rice. We call na basmati rice, Indian gate rice, all these. Flavor, savor, tomato. The tomato can withstand for longer time, right? Which is PG encoding gene, polygalactose encoding gene. Herbicide tolerance, which is glyphosate. This is also there in the slides of the secondary metabolites. Right? Simple. This also ATPase sir explained. Fish is a technique which is used to identify the mutations, especially to identify the locations. Okay, that is the fish. So this is also 10th standard question only. Wavelength of the light used and numerical aperture. The common thing. This is good question. Human embryonic lung. At least you know one HeLa. HeLa is what? What it is? HeLa, what cells there? MHCs. Correct na? Tell me what is the answer? Sorry. Correct. Tell me, what is the answer? Human embryonic lung. See, MRC5 is a human fetal lung fibroblast line. HEK represents kidney, HeLa cervical carcinoma. Okay. HLA is MHC, human leukocyte antigen. Right. Then VISH, which is representing amniotic fluid or amniotic cavity. This is also sir explained. So simple it is. SDS phase. Correct na? SDS phase is generally a method which is used to separate the DNA or proteins based on their sizes, right? And we know that larger fragments move slowly, smaller fragments move very faster on a gel medium towards with their respective electrodes. Now, very important question, tell me, the point I want you to understand here, what we can say. Suppose example, I am separating the DNA. I kept the DNA on the gel plate. DNA moves towards which electrode? Anode or cathode? Every one of you answer. Suppose if I keep the DNA moves towards which electrode? DNA is what charged? Negatively charged. Obviously it starts moving towards what? Positively charged. Right? Depending on the charge only they will move. These are all you read in your masters. See, this you have to remember. Trust my words, again there is a possibility that you can get this question. What is the answer for this? It's also straightforward question. I really don't know except this ANOVA chi-square and jet test and Kruskal-Wallis test, which is used to compare the relationship between more than two population groups. And what about the jet test? Jet test is very simple. We use it to calculate the mean differences. This is the right. ANOVA having multiple applications. ANOVA we can use. ANOVA, what is the full form of ANOVA? Analysis of variance. Right. We can use in the drug analysis. We can use in the protein analysis also. Right. These are all statistical tests which is used to measure the differences, right? So you just remember ANOVA is analysis of variance. What is the most appropriate answer here? Option A will be the 2. B will be Z test we used to calculate the mean. ANOVA, analysis of variance it is. Correct na? And what is the option? Comparing means of more than 2 population groups, right? Chi-square test? Right. It is always talk about the fit, right? Answer is 1. Sir explained this also, AFLP in one single class. Of course, AFLP is a PCR based tool where we generally use the restriction enzymes to cut the specific location on the DNA, followed by the ligation, then our desired fragment we will do the amplification. Right. Freshly prepared. See, that is also a question. See, remember all the answers are correct. What is the significance of X-ray crystallography? Why we used this? Everyone answer, what is the significance of the X-ray crystallography? See, X-ray crystallography is a technique which is used to understand the structure of the proteins by understanding atoms and their positions, and thus X-ray crystallography exclusively dependent on the principle which is Bragg's law, which is used in determining arrangement of atoms within a crystal, right, which is a non-destructive technique which follows the elastic scattering of X-rays. All 4 options are right. See, at least out of 100, more than 85 questions just direct and straightforward questions only, okay. But compared to the 2024, 2023 is little qualitative paper, okay. That we will discuss in the next class, okay. Tomorrow morning everyone attend without fail. I will inform the timing prior, either 8 or 8:30 I will take, okay. Because net now it takes a lot of time to solve that paper, okay. Now what you do is, use the same paper and revise the entire paper one more time, because trust my words, the same statement will be reappearing in your exam. That's why 4 paper, 23 paper, 22 paper and 21 paper. You try to repeat these 4 papers only 2 to 3 times, because trust my words, same lines will be repeated or the similar type of questions only will appear, right. So in the morning we will start this, okay. The NET 2025 July paper we will start. Clear everyone online? Because there is no great concepts are involved here, because it's all are statement based only. Clear everyone, revise one more time online people. Now we'll see you in the next class tomorrow morning. Everyone felt bored because of the slow class. That is common.